organism needs to respond to its environment. It needs to be able to change, to detect things coming in and respond appropriately. So how might that happen at the genetic level, at the transcriptional level? Well, many things are a balance in nature, and transcription is also probably a balance of RNA polymerases going in one direction, and when they're making a protein from that RNA, that's called messenger RNA, and that's defined as sense direction. But it turns out there's a great deal of antisense transcription taking place as well. And some of the most basic early decisions in a cell differentiating, in fact, is trying to decide, is the RNA polymerase 2 going to go in a sense direction or an antisense direction? But it's possible that there is a balance, that it's both. And that balance can shift and change depending on what the organism or the cell itself might need to do. And it turns out that there was this terrific experiment done by Noelle Latoile at the University of California in San Francisco, and about 3 years ago now. But it was very clever because she took these very basic organisms, they're little worms called C. elegans, and they move towards food if they sense something, and it's called an odor, and it's they will sense a molecule, and they will move toward it. And she experimented and played around with them, and there's some chemical you can give where at first it's very attractive, and they move toward it, but then they realize it's a little toxic and they don't like it after all. And actually, they learn over a short period of time not to move toward that molecule. And that's called adaptation or the development of tolerance, and it's reversible. So you can measure different things that go on. And it turns out that she discovered that during that period of time that the worm was learning not to go toward that bad smell, it was making endo-siRNAs to the actual gene that it was responding to the odor in the first place. So it's actually called an odor gene, or odor 1, or odor 2, or something. And odor, the messenger RNA, was beginning to go down as the organism learned not to go in that direction. And at the same time, these endo-siRNAs were being formed that were specific for that messenger RNA. So these endo-siRNAs were being recognized by the Argonaute protein and were carrying transcription inhibiting or translation inhibiting protein complexes around with them, and they would interact with the messenger RNA and decrease it. So you had less and less of this protein being made, so less and less urge for that worm to move toward that odor. So the worm stopped moving. It had adapted to its environment. So the question is, why was that messenger RNA going down? And then, in fact, where was that endo-siRNA coming from in the first place? So the idea is here, I'm thinking if there's a balance between transcription and sense direction and antisense direction, and if those two things come together and you have a transcript being made in sense and in antisense, and you get that kissing stem loop, the interaction of a cis, something being made in cis that's an antisense, nascent antisense transcript. If you had that taking place and you had the development of a double-stranded piece of RNA from these two single strands, that would be a source for your endo-siRNA. So that transcription in one direction from the very same gene leads to the increase in the movement towards the odor. Transcription in the reverse direction literally causes the animal to stop, to reverse its direction. Simply because you have triggered, for some reason, bilateral transcription that is in the other direction and becomes more and more strong in the other direction. So why can that happen? Well, there's also these things called cryptic promoters, which are just like regular promoters, but they're hidden, they're cryptic. And these promoters are triggered to make these cis nascent antisense transcripts. And here, I can't tell you exactly how. I can only tell you that often cryptic promoters are part of a retrotransposable element. The long terminal repeat of a retrotransposable element is nothing more than a promoter and enhancer. And if you had this in an upside down direction, if it had transposed there way ancient time in history, it was turned off for most of the period of time as the cell differentiates and things are turned on and off and decisions are made. So it's usually turned off. But if it gets turned on, you've got endo-siRNA, and you can have adaptation to the environment.
And there's a term that you're thinking of?
Oh, I just call that self-pseudogenes. Because in a sense you're making a gene against yourself. It's a pseudogene against that very gene itself, from that very same place. Usually pseudogenes are in two different places, but hey, you can be a pseudogene to yourself.